my great pleasure to introduce Dr. Andrew Bellizzi. He's a clinical professor in the Department of Pathology at the University of Iowa, and he directs both immunopathology lab and the GI pathology as well as the GI fellowship program. Uh, I met Andrew briefly while I was a trainee at Brigham Women's Hospital, and he was a junior faculty there. And over the years, Andrew has really become the true expert and leader on the immunohistochemistry field. He's the past president of the CUP immunohistochemistry subcommittee. And as he sometimes calls himself the IHC concierge. So it's a really if you have any question about any marker, this is something I really admire him. If you have any question, any biomarker, I don't think he basically knows every biomarker is incredible. So if you have questions, it's probably a good time to chat with him later after the talk. I'll catch him later. So his talk today is next generation immunohistochemistry, how to build highly effective diagnostic tests in AP service, which is really exactly what we want to hear. So Andrew, thank you so much for coming, and uh, you may get started.
All right, thank you. Thank you so much. That what a wonderful introduction, and I really appreciate how personal it was, and it's really apt. I'm so deeply passionate about immunohistochemistry, and it's a tool that I've used over the course of my career to explore oncopathology and really all of oncopathology, even though I practice as a GI pathology subspecialty expert. I'm broadly interested uh, in all of oncopathology, including hematopathology, sarcoma pathology, et cetera, et cetera. And immunohistochemistry is the thing that ties me to all these areas of subspecialty expertise. And I I don't know. It might show up in the next couple slides or two. I offer my services as an immunohistochemistry concierge because one piece of advice for junior people is do what you love and really lean into it. And one thing I say is if you say you're the expert, you're the expert. And the way you develop expertise is have interest and know a little bit more than other people, but most importantly, offer your services as a helper. And people posed me the most interesting questions, and through researching people's problems, I've developed really pretty profound expertise in the area of immunohistochemistry and biomarkers. All right, I'll stop yammering and show you a few PowerPoint slides. Can you guys see the PowerPoint slides?
Yes,
Great.
yes, it's perfect.
All right, thank you. So far, I'm not able to advance my slides. You'd think after all these years on PowerPoint, I'm gonna unshare.
Try clicking on the PowerPoint. Sometimes when you share,
Yeah.
you're actually on Zoom and not on the PowerPoint itself.
PowerPoint. You still see my slides? Huh.
You'll
Yes,
see it.
yeah.
Yep.
Great.
And there it goes moving.
Let's see.
It's moved to your disclosures.
Yep, got it. Thank you. Thank you. I've got one disclosure. I took a flight and a hotel and registration fees for the CAP annual meeting from AstraZeneca to give a low HER2 pan HER2 talk. And the day before I was going to give the talk, it got canceled, and now I have to disclose that for the next 2 years. Uh, Shameless plugs. One way to connect, the best way to connect with me is by email, andrew-bellizzi@uiowa.edu. But after that, I'm very active on social media as IHC_guy. And I share a lot of educational content pertaining to immunohistochemistry, and I interact with folks asking questions or asking about technical aspects of immunohistochemistry. I also would shout out ISOM, the International Society for Immunohistochemistry and Molecular Morphology, which is the professional society devoted to immunohistochemistry, the journal AIM, Applied Immunohistochemistry and Molecular Morphology, which is affiliated with ISOM. I have a pathology podcast at PathPod, and shout out to my home institution at UI Pathology. A little bit more about ISOM. ISOM's principal intellectual output over the last 5 years has been a series of webinars that are monthly, always Wednesday, always mid-month, always 11 a.m. Eastern time. And they are recorded and they're posted to the internet after the fact. So they're available at this web address. And the next one's in a couple weeks. Dr. Christina Maggie Galuzzi is going to discuss immunohistochemistry in the differential diagnosis of kidney tumors. Most of the talks are end-use applications delivered by uber subspecialty experts, and you're welcome to join for the webinars. All the educational content is free, and if you're interested in hearing more about that, hit up the web address or shoot me an email. So here's the elevator pitch for the talk. I can build an immunohistochemical test to virtually any target. And the value proposition is at the end of this lecture, you'll have the tools to build an immunohistochemical test to any target. One of the most frequently asked questions when I bring a new marker to the meeting is, are these reagents commercially available? And I'll only build a test from reagents that are commercially available. And there are dozens, if not hundreds, of primary antibodies to virtually any protein, because immunohistochemistry is fundamental to science, and science is so well funded that there's an ecosystem where there are gobs and gobs of reagents available. Everybody benefits from smart, technically sound immunohistochemistry, and there it is. I'll be your immunohistochemistry concierge, and I'd love to talk to all kinds of folks. I like to talk to trainees. I love talking shop with IHC lab directors, clinical faculty, and scientists. You know, immunohistochemistry is fundamental to the work that we do diagnostically every day. I'll also mention for trainees and clinical faculty. Yeah, I sought out the role as an IHC lab director because I thought as IHC lab director, I could most deeply, positively impact the work of my entire group. But it's also served me incredibly in my research career because if you have talent for immunohistochemistry, you can do so many things. Like the research that I do, I hope you'll find to be very accessible and a model for a way to have a research career. So the outline for the talk, I'm going to briefly talk about the proposed regulation, FDA regulation or oversight of laboratory developed tests that thankfully was recently struck down. We'll highlight a couple key immunohistochemical principles. I'll introduce this concept of next generation immunohistochemistry that my academic career is largely based on. and then we'll take you through the processes of selection, optimization, and validation. And then I'll take you through the process for one marker. I I've given a form of this talk for a number of years, and I used to take the audience through all of my research output. These are various what I call next generation immunohistochemical markers that I've quote unquote discovered over the last several years, and I used to take you through how I selected and validated OTC as our most sensitive marker of hepatocellular differentiation, IGF-2 as a marker of paraganglioma, pheochromocytoma, et cetera, et cetera. And after giving the talk for several years and the list kept getting longer and longer, I thought it'd be more helpful to take you through the process rather than the product. In terms of FDA oversight of laboratory developed tests, which impacts all of laboratory medicine, but deeply impacted me in immunohistochemistry. In my 250 immunohistochemical test menu, I have 249 laboratory developed tests and one FDA approved assay. The FDA was asserting regulatory oversight over laboratories and was holding individual laboratories to the standard of manufacturers. And this final rule was strongly opposed by many thought leaders in pathology, including AMP and ACLA, the American Clinical Laboratory Association, filed a lawsuit and won. And the final rule was overturned, and we'll see what happens in the vacuum. For my entire career in immunohistochemistry that spans a couple decades, the FDA this has come and gone in waves. The FDA has, I say, threatened to assert regulatory oversight of laboratory developed tests over the course of my entire career. And of course, this could be the subject of an entire hour's webinar. I mentioned that I target my education to all kinds of learners, including basic scientists and physician scientists. And these are two references that I specifically highlight for basic and physician scientists that describe on the left what can go wrong with immunohistochemistry in basic science, and on the right, a proposal for what constitutes quote unquote validation of antibodies in the basic scientists. And I'll compare and contrast this with what validation looks like in the clinical space later on in the talk. First key, and really most important, key immunohistochemical concept that came around after I'd been in the field for maybe a decade, and that's fit for purpose. And that's the concept that immunohistochemical assays should be conceived, optimized, and validated to meet a specific purpose, and that a single immunohistochemical assay, a single set of conditions on a single autostainer may be poorly positioned to meet multiple purposes. And the idea here is we tend to think about immunohistochemical tests in an analyte specific way, and it's more productive to think about them from a diagnostic or predictive application way, because the way that IHC tests are optimized, they perform ideally in a generally narrow range of analyte concentration. And in different diagnostic applications, the analyte of interest may be barely present or or they're in spades, and an assay optimized for a barely present antigen will perform potentially poorly in another condition in which the antigen or analyte is present in spades, and vice versa. The most famous example uh, is ALK, where we have assay the traditional original ALK1 clone was designed and fit for the purpose of detecting ALK expression in anaplastic large cell lymphoma, where ALK is very present, and assays built on ALK1 perform poorly in the detection of EML4 ALK rearranged lung cancers, where that fusion protein is barely present. I'll briefly take us through another omnipresent example, and that's ER clinical immunohistochemistry assays. And around this fit for purpose concept, you can conceive of a few purposes. One, we can use ER assays to select patients for endocrine therapy. That's a predictive purpose. And it's in bold there because that's the principal purpose of ER clinical assays. But you could also think about ER clinical assays in the distinction of luminal breast cancers from triple negative breast cancers. That's a diagnostic application. Or to predict survival, that's a prognostic application. And I'll argue that contemporary and for the last couple decades, ER clinical immunohistochemical assays are optimized for this first purpose and thus perform suboptimally, I should say, to poorly for the second and third purpose. And this is a combination of data from two studies. We've got black bars and blue bars, and the black bars are from a study from Laura Collins and Stu Schnitt, and the blue bars are from a study by Harvey et al. The black bars reflect the performance of contemporary immunohistochemical assays for ER, and the blue bars reflect the performance of really first generation initial assays developed to detect estrogen receptor in breast cancer. And Collins and Schnitt in the black bars found that the results of ER immunohistochemical stains are entirely dichotomized, that tumors were either entirely negative in your 10, 15, 20% of ER negative breast cancers, or were, I say, whiz bang positive or coat sleeve brown or blammo, they're diffusely strongly positive. And they were a bit confused about that because in these first generation assays, there were negative cases, a similar proportion of negative cases, but the positive cases, the ER results were largely normally distributed. And something happened between this publication in 1999 and this publication in 2005, and there were technical advances in detection chemistry, uh, primary antibody, and then secondary antibody, aka detection chemistry. And the detection chemistries that were developed, they're referred to as polymer based, are a signal amplifying technique. And what resulted, the output was the dichotomization of a continuous variable. So in this publication from Harvey, the skinny blue bars one, uh, historically, with historical immunohistochemical assays, they did a really, really good job of stratifying patient survival. But with the dichotomization of this continuous variable, we lost that purpose. So advantages, problems, and challenges, and the promise of immunohistochemistry. everybody's familiar with the advantages. Immunohistochemistry is cheap. It's widely available. I'm emphasizing for folks interested in developing laboratory developed tests that we've got an endless supply of commercially available primary antibodies. The few main problems, well, one, there's no formal training in IHC lab directorship, and that's one of the prime drivers for me teaching in this space and offering my service as a mentor and wayfinder. All my fellow IHC lab directors that are seasoned like me have learned on the job, though I will say we have a very welcoming community and support each other tremendously. Running an IHC lab, uh, consistently resourcing high quality control is the greatest challenge that I continue to face. And in my former role as the chair of the Immunohistochemistry Committee, overseeing our national proficiency testing program, uh, consistently resourcing high quality material for EQA, external quality assessment, synonym for proficiency testing, was my greatest anxiety. Another problem, challenge, and maybe misconception is that because of this signal amplification from the polymer-based detection that I referred to, immunohistochemistry isn't quantitative, rather it's ordinal, and we'll talk more about that. The promise, and I'm not going to talk very much about predictive marker immunohistochemistry, but diagnostic immunohistochemistry, it's mainly useful to know positive, negative, weak, moderate, strong, but for contemporary predictive marker assays, we need more precision and accuracy. And we've got some new tools to, I say, look under the hood around the performance of immunohistochemistry, including isogenic cell lines characterized by mass spec and glass beads that are coated with known quantities of analyte, and we can glean more information about the analytic performance of immunohistochemical assays using this quantitative reference material that's only become commercially available in the last few years. And I use both of these in my lab for optimization of predictive marker immunohistochemistry and for continuous quality control. And this is, I say, toward a standardized immunohistochemistry. And I'll shout out my friend mentor David Rim, whose career is based in quantification of immunohistochemical analytes and standardized immunohistochemistry. Array science, here's my friend Regan Fulton in the middle there. He has a company that's devoted to producing these cell line microarrays that I find to be incredibly valuable in the quality control of my lab. Here's a product that he produces for MMR immunohistochemistry. It's isogenic cell lines in which he's got a cell line that's CRISPR-Cas knocked out separately, MLH1, a separate cell line, PMS2, a separate cell line, MSH2, a separate cell line, MSH6. I wouldn't be comfortable controlling my mismatch repair immunohistochemistry without access to this material. And then here's the glass microbeads, our product of Boston Cell Standards. It's a really, really interesting group. The founder CEO is Steve Bogan, and he's a clinical chemist. And one of the concepts, one of my general concepts teaching anybody is a superpower is to be able to apply domain expertise from one domain to your own domain, and using concepts from clinical chemistry has allowed us to advance the field of predictive marker immunohistochemistry uh, around standardization and quantification. And I would highly recommend this review article published a number of years ago, a root cause analysis into the high error rate in clinical immunohistochemistry. And here's a schematic of his microbeads. Steve has gotten funding. I didn't mention it, but I said that immunohistochemistry is science and science is highly funded, but that ironically the science of immunohistochemistry is hardly funded at all. And here's an exception. This CASI, Consortium for Analytic Standardization in Immunohistochemistry, is an effort to, again, look under the hood of the performance of immunohistochemistry at laboratories toward standardization. And the first CASI paper that describes the analytic performance of HER2 immunohistochemistry around the world is in press in The Lancet. And this is what you can do with the cell lines and the glass microbeads. You can develop analytic response curves to immunohistochemical assays, and these analytic response curves relate the analyte concentration to the stain intensity. And we've got four analytic response curves here. These are four analytic response curves generated to four different FDA-approved PDL1 assays. Analytic response curves that are shifted to the left show more signal for a given analyte concentration, and analytic response curves shifted to the right are characteristic of assays that show less signal for a given quantity of analyte. And if you look at these curves, you see one of these curves is not like the other. And if you're familiar with PDL1 immunohistochemistry, the right shifted analytic response curve, the less signal for given quantity of analyte is for the SP142 assay. And this is very insightful and explains the clinical performance of SP142 in the field. And I'll come back to this as sort of interest dictates. And I've begun to use this material in my laboratory. I mentioned its use in optimization and on-slide quality control. This is a series of experiments that I performed to demonstrate the interchangeability of our PDL1 assay on the four immunohistochemistry analyzers in our laboratory. So that's the serious part. Onto the fun. Next generation immunohistochemistry. And my approach to immunohistochemistry is it's an applied science, and the source of the markers that I want to develop are their derivation is from molecular genetic discovery and developmental biology. So I'm looking for protein correlates of molecular genetic events and lineage restricted transcription factors. And we might identify these lineage restricted transcription factors in the context of gene expression profiling experiments. And our tests keep getting better and better and better. So this is the kind of data that drives me wild. This is the initial description of GATA3, actually, as a marker of bladder cancer based on gene expression profiling that John Higgins and Matt Van der Rijn did in GU tumors, in kidney, bladder, prostate cancers, and identified GATA3 as highly expressed by urothelial carcinoma. Here's an example of uh, what I refer to as a tumor type being solved. So this is a paper that described the underlying molecular genetic event, the oncogenic driver of solitary fibrous tumor. It's NAB2 STAT6 fusion. And anytime the molecular genetic underpinnings of a tumor type is solved, it begs for the development of a next generation immunohistochemical assay. And so within a few months of this paper being published in Nature Genetics, Leona, my former resident fellow, and Jason et al. Published this paper on STAT6, which is a nearly perfect immunohistochemical marker for solitary fibrous tumor. Historically, our immunohistochemical armamentarium was empirically derived. We used to chop up cells and tissues and cell lines and generate a panoply of monoclonal or polyclonal antibodies of unknown specificity, and then we would try to figure out what protein they were labeling, and we ended up with markers like mammoglobin and growth cystic disease, fluid protein, and in general, cytoplasmic and membranous differentiate, I call them differentiation markers, tend to be expressed in a rare cells, weak, patchy, I say wifty, wafty manner in poorly differentiated tumors. That is, their expression, the amount of expression tends to be inversely related to the differentiation of the tumor, as opposed to lineage restricted transcription factors. This has got a three in the same breast cancer. And I refer to the primacy of lineage restricted transcription factors. In general, lineage restricted transcription factors are best. I think about new immunohistochemical tests as technologies, and technologies can be experienced by us, interpreted, described in the context of this Gartner hype cycle. Uh, New technologies often come with an inflated expectations. Their real world performance generally doesn't line up with initial expectations. And eventually we learn more, we understand more, and we have this more realistic application or plateau of productivity. My friend Alan Gown often, I've heard him say that the best time to use an immunohistochemical marker is right after it's published, before all its warts have not been revealed. But I actually have a different view, and my view is that the Gartner hype cycle that we experience with new immunohistochemical analytes is largely a function of poorly designed experiments. Generally, new immunohistochemical markers are described and test studied in the context of a single diagnostic application. And think back to this fit for purpose concept. And instead of starting here, going here, and ending up here, I argue that, well, we should demonstrate all the potential diagnostic utilities of an immunohistochemical marker when it's initially published. And Markku Miettinen is the best at this, and he's been a North Star for me over the course of my career. He doesn't just describe the performance of an immunohistochemical marker, he bombs it. He describes highly effective immunohistochemical markers in thousands of tumors of dozens of types. And over my 15 years at Iowa, I've emulated him. And he works in multi-tissue blocks. I work in tissue microarray, and I've tried to recapitulate his work to be able to describe new immunohistochemical tests in multiple diagnostic contexts, really in pan-cancer diagnostic concepts. And the observation is that nearly all transcription factors are oligospecific rather than monospecific. And I use this concept of threat opportunity. If you're only familiar with a single diagnostic application for a marker and you apply it and it's positive, then you can only make that diagnosis. But if you're familiar with the frequently multiple diagnostic utilities for immunohistochemical markers, you can use them in really refined manners. And so Marku and I are doing extensive diagnostic evaluation, but I also do what I refer to as in silico visualization of gene expression. I can predict the results of Marku's experiments to a large extent, or my own, before I perform the experiments using a few tools that I'll share with you in the next few minutes. So stains I love tend to be oligospecific transcription factors, and this is data from one of Marku's papers. Most folks are familiar with GATA3 as a marker of bladder and breast cancer, but Marku showed that it was expressed by 8 or 9 different tumor types. And threat opportunity, if you're familiar with this, you can use GATA3 in a gajillion different ways. So you can use it, and lung pathologists are very savvy to this, in the mesothelioma versus lung adenocarcinoma differential diagnosis, or I use it not as a mainline diagnostic marker, but as adjunct support in the pancreatic adenocarcinoma versus other GI adenocarcinoma differential diagnosis, etc. I've I've done similar work with a marker SATB2. SATB2 was initially introduced to the pathology community in this paper. That was the output of the Human Protein Atlas, and SATB2 was identified as a marker of colon cancer. I think one of the most important take homes from the talk is that immunohistochemistry is science, and immunohistochemistry for transcription factors is developmental biology, and every new immunohistochemical marker is a gene that's been cloned generally decades ago and has decades of research experience in the developmental biology literature. So SATB2 was introduced only as a colon cancer marker, but there was a rich developmental biology as about around SATB2 as a marker of skeletal development and craniofacial patterning. And so SATB2 is also a marker of osteoblastic lineage, and SATB2 has a number of diagnostic applications. It's a lower GI specific marker. It's more sensitive than CDX2 as a marker of medullary carcinoma of the colon, or undifferentiated carcinoma of the colon. Here it is functioning as an osteoblast lineage marker. I'm a neuroendocrine tumor pathologist among other identities, so I've explored SATB2 in my patient cohorts and have found it to be a marker of rectal and appendiceal neuroendocrine tumors. In neuroendocrine carcinomas, it's a marker of Merkel cell carcinoma. And in small round cell sarcomas, it's a marker of B-core rearranged sarcoma. And so I always ask, will it Swiss Army knife? And the answer for this one is emphatically yes. All right. So on to the optimization, validation, and selection optimization and validation. I spend a lot of time mentoring IHC lab directors, and the advice that I give is largely dictated by the analyte of interest. So here's my friend Frank Ingram DMing me about wanting to bring up GATA3, Pax8, SOX10, or I call these tried and trues. And here's my friend Phil. He's the IHC lab director at OHSU in Portland. And he was asking about a very niche application. He was looking for advice on toxoplasma immunohistochemistry. And this is a simple slide, but it's one of, if not the most important slide in the talk. And this is where I go for sources of primary antibodies. And at the top is phone a friend, and I use that for esoteric antibodies. So for Phil, the advice for the toxoplasma was phone a friend. We look in the published literature, but for what I referred to in the last slide as tried and trues, and what I refer to here as workhorses, the Nordic QC website, which is the Scandinavian EQA PT program, is the single best source of not just primary antibodies, but autostainer platform specific protocols. They publish incredibly detail specific uh, results of their proficiency testing scheme. And then for folks that are interested in research, here are three databases that I use to identify quote unquote novel antibodies for research. BioCompare, which is what I initially used, which is just kind of a dumb database, but Antibodypedia and BenchSci, which I will show you, which are much more sophisticated. So here's a screenshot from Nordic QC. You can find, depending on your analyte of interest, HER2 platform, DACO autostainer link 48 versus Omnis versus Bond 3 versus Benchmark Ultra, platform specific protocols that you can click on PDF. And all this information is free. Here's the BioCompare Antibodypedia. The advantage of Antibodypedia is BioCompare will just spit out a list of antibodies. Antibodypedia will vouch for the quality of the science behind the validation of the primary antibodies, and you're looking for green circles or green stars. You're looking for vendors that have done enhanced validation of the immunohistochemical markers, and this is validation in the basic science sense, and we'll come back to that in just a second. And then here's BenchSci. The advantage of BenchSci is that it links to vendors, but it also links to the primary literature. I used to PubMed, and NCOA2 is the protein of interest here. I'd PubMed NCOA2 and immunohistochemistry and pull up all the papers and download the PDFs and read the papers and look at the figures. And BenchSci does that for you. It links to the papers directly, and it shows the germane figures directly. So this saves me gobs and gobs of time. And both BioCompare, Antibodypedia, BenchSci, these are all freely available. Ethical sourcing of primary antibodies. I won't use Santa Cruz antibody in my lab. Santa Cruz was here's another lawsuit, right? Santa Cruz was found guilty of gross animal rights violations and lost their ability to be purveyor of animal products. And the only reason that they're still in business is the US FDA or USDA does not consider mice to be animals. So they went from a rabbit, goat, polyclonal, extensively producing company with some mouse monoclonal on the side to an exclusively mouse monoclonal producing company. Components of an immunohistochemical assay, what I refer to as the knobs and dials, and this is an introduction to optimization of immunohistochemical assays. So we pay attention to antigen retrieval, whether it's pH, usually pH, lower high pH, or enzymatic. We can manipulate the temperature and the time. The primary antibody, of course, is very important, monoclonal, polyclonal. We're interested in the specific clone, the vendor, the dilution, the primary antibody incubation duration, and whether we incubate at room temperature or heat, and the detection chemistry, which I highlighted in the ER clinical immunohistochemical assay slide, where we flexed from traditional detection chemistry to this polymer-based detection chemistry. And all of these components of the IHC assay can be manipulated to ultimately modify the shape and the left or right shiftedness of the analytic response curve. Optimization describes the series of experiments that we perform to lock in antibody conditions that we want to use for the validation. And here's an example of a data sheet. This is from my lab notebook that I use for IHC optimization. So here's the analyte of interest, the vendor, the tissue type that I'm performing the optimization in, and the description of the initial series of experiments. So different dilutions, different retrievals, no antigen retrieval, high pH retrieval, low pH retrieval. And I iteratively experiment, modifying conditions to mainly turn up or turn down the signal until I think it's just right, and then we proceed with validation. In validation, in the clinical lab, validation refers to the examination of a prescribed number of tissues, of expected positive and expected negative. There are, based on this CAP Center guideline, expectations for the performance of the antibody in the validation. There are different recommended prescribed sizes of cohorts for diagnostic versus predictive assays, with the idea that predictive assays are more serious and potentially poorly optimized validated assays more dangerous, and that they deserve further scrutiny. We used to have different prescriptions for specifically call outs for ER and HER2 based on whether the assay that was being validated was FDA approved or laboratory developed. I participated in a guideline update with this wonderful and erudite group, and we made a bunch of changes. One is introduction of the indirect clinical validation for predictive markers without an orthogonal gold standard, which is just most contemporary predictive markers lack an orthogonal gold standard, which is incredibly problematic. And I'll leave that up there for just a second. But this is essential reading for any IHC lab director, as the CAP anatomic pathology checklist is largely derived from this guideline. So validation in the research setting is mainly around specificity. And in the research setting, we do science to determine the specificity of an antibody. So we do genetic modification of the target of interest to see that the band on the western goes away, or we correlate with the results of mass spec. One accessible type of validation is if antibodies to an analyte recognize different epitopes of the analyte of interest, and they produce the same result in the same set of cells or tissues, that gives us confidence in the specificity of the antibody. Most all clinical laboratories don't have access to tools for this type of validation, and we rely on the vendors having done this validation. We're able to validate the diagnostic performance of immunohistochemistry in the me and Marcoux way, where we examine a bunch of tissues. We're really good at tissue type, analyte of interest, sensitivity, specificity, positive predictive value, negative predictive value. All right. We're in the home stretch. This is the grand finale where we run through the selection, optimization, and validation of a novel immunohistochemical marker using all the tools and tricks that I've shared with you for the first 45 minutes here. And I I tweet about this stuff, and I frequently tweet my selections, optimizations, and validations. And so there'll be a couple Twitter screen captures here. And here's a peek behind the curtain. We'll come back to this, but this is the immunohistochemical stain that I'm going to describe. And for any immunohistochemical stain, because I'm so clinically grounded, it's always, what diagnostic problem am I trying to solve? What are the currently deployed diagnostics? I don't make a test to make a test. I make a test because the current tests are imperfect. I try to make a test where a test does not exist, or a test that's manifestly superior to the currently deployed diagnostics. So what are the limitations of the currently deployed diagnostics? And then how did I find the target? Uh, what's the target, and can you envision extended utilities? Because remember, I love these oligospecific transcription factors. So what diagnostic problem am I trying to solve? Uh, it's a protein expressed in the nucleus. It's probably a lineage restricted transcription factor. This is a screenshot of normal pancreas, and we've got islets of Langerhans are negative, and some ducts are negative, and the acinar cells are uh, strongly positive. And I say this is a very important teaching point. Like, residents, trainees, prick up your ears. Tumors tend to recapitulate cells native to the organ in which they arise. So I've got an immunohistochemical marker that's positive in acinar cells, and so I'm trying to make a diagnostic for acinar cell carcinoma. Little bit on acinar cell carcinoma. Here's a couple images of acinar cell carcinoma. Tends to have uniformly prominent nucleoli. If cases of acinar cell carcinoma are misdiagnosed, they tend to be misdiagnosed. They're frequently misdiagnosed as neuroendocrine tumors, occasionally misdiagnosed as ductal adenocarcinoma. And the mainline diagnostic for acinar cell carcinoma is trypsin immunohistochemistry. And there are a few others. People use BCL10, but immunohistochemistry is science. acinar cell carcinoma, this BCL10 antibody, this specific clone from this naughty vendor. It's actually a antibody that recognizes the carboxy terminus of BCL10. And in acinar cell carcinoma, it's not reacting with BCL10. It's actually reacting with carboxyl ester hydrolase, which is a pancreatic exocrine enzyme. There's some molecular genetics, but there's not a single oncogenic driver. BRAF fusions are common, but they're only seen in 20%. So it's mainly trypsin. Folks use BCL10. I won't use BCL10 because of the vendor. And then trypsin's great, except I said, if you're the expert, you say that you're the expert. If you make yourself available as an expert, people present you the most interesting problems repeatedly over the course of my career. Here's an email from Sarah, IHC lab director at UCSF. This is from Audrey Lazenby, long tenured AP director at UNMC in Omaha. Lots of labs struggle for trypsin, and that's how I derive my research inspiration. We've got an important diagnosis, challenging differential diagnosis with a mainline diagnostic that people are struggling with. And if people are struggling with trypsin, I'm going to try to build my own better, best test. And I started here. I started with PTF1 alpha, which is a transcription factor that tells an epithelial cell in the pancreas to be an acinar cell. And I tried a few years ago, I tried initially a monoclonal and a polyclonal, and I couldn't get either of these antibodies to react. And then how do I identify immunohistochemical targets? Uh, it's not rocket science, it's Google. A common query is what transcription factors regulate, in this case, pancreatic acinar development. Here's the top hit. It's this PTF1 alpha. But if you scroll down to maybe hit 10, 15, you end up at this marker MIST1. And I had asked on Twitter if anybody could suss this out, and one of my friends, Hubert, guessed that it was MIST1, although he considered the possibility of PTF1 alpha. Next generation immunohistochemistry is translational science. I gave grand rounds at Yale a few years ago, and I bumped into I had a meeting with Wanjie He, who studied MIST1 in his postdoc, and I had read his papers as I was developing this immunohistochemical marker, thinking about this as a target. And Wanjie showed that MIST1 regulates the size, shape, and polarization of secretory granules and secretory cells. He specifically studied it in the gastric corpus. It's expressed by chief cells. But I asked him if he thought it would be a decent marker for acinar cell carcinoma, and he said, I wish I'd thought of that. So how to visualize gene expression, and I'm looking at the clock. I see we're at the top of the hour. I'm gonna bring this to a grand finale in the next two to 3 minutes. Uh, how to visualize gene expression. Any gene you're interested in, I'd hit up GTX, Illumina, or BioGPS, but the first place I go to learn about a new gene protein is Wikipedia. This is the GTX for MIST1, and it's off the charts in pancreas, and that validates the initial image that I showed you of MIST1 expression in the pancreas. How do I visualize gene expression and molecular genetics in cancer? I use this database called C Bioportal. which is incredibly user-friendly and web-based. The problem with gene express using cBioPortal for gene expression, it only largely includes TCGA tumor types, and acinar cell carcinoma isn't in the database. So I suspect that for acinar cell carcinoma, the gene expression would be off the charts. Here's I should say, optimization for MIST1. And here's the results of the initial experiments. I found it to be a sensitive and specific marker in this differential diagnosis of pancreatic tumors, but I was bummed at the signal to noise ratio. I wanted more signal. Uh, and Hubert said, interesting that it's weaker than in non-neoplastic acinar cells. Is this expected? And I'd say not entirely unexpected. I bragged about the primacy of lineage-restricted transcription factors, but here's SATV2 in a rectal neuroendocrine tumor relative to the overlying rectal mucosa. And even though transcription factors are robustly expressed in tumors, they tend to be somewhat less strongly expressed. And in my haste to get this stain up and running, I made the poor choice of optimizing the test in benign pancreas and normal acinar parenchyma when I should have used an acinar cell carcinoma. So here's my initial assay. And then I said knobs and dials. So I say, stronger, longer. And I went from 1 to 400 to 1 to 100, so 4 times more concentrated. I lengthened the primary antibody incubation. And what I really did was for the analytic response curve for this assay, I shifted it to the left. And then the rest is academic. So MIST1 positivity supports a diagnosis of acinar cell carcinoma. It's essentially 100% sensitive and specific. And then around additional potential diagnostic utilities, here it is in the gastric body. Here it is. This was especially exciting. Here it is in the parotid. It's also expressed by plasma cells. Plasma cells are the acinar cell of the hematolymphoid system. Uh, and there was a two papers that were previously published in the diagnostic literature around MIST1, and one showed that it was a marker of a cynic cell carcinoma of the salivary gland, and one showed that it was a marker of plasmacytic differentiation. So here's another diagnostic application. Diffuse strong staining obscuring nuclear detail supports a diagnosis of a cynic cell carcinoma. Sort of heterogeneous, but fairly strong staining supports a diagnosis of acinar cell carcinoma, of secretory carcinoma, excuse me, and otherwise it's not significantly expressed in other salivary gland tumors. And then here's the final diagnostic application. It's a wonderful marker of plasmacytic differentiation, and I use it in my GI pathology work as a marker of plasma cells. So ultimately, MIST1 will its Swiss Army knife, yes, and I use this marker in my lab for the diagnosis of acinar cell carcinoma, acinic cell carcinoma, and plasmacytic differentiation. But really, just an example of the primacy of lineage-restricted transcription factors and immunohistochemistry is science. And that's all I got. Thank you so much for the invitation. Thank you for everybody's time and attention. And thanks for letting me share my joy for pathology and immunohistochemistry with you. Don't forget fit for purpose. Immunohistochemistry is a tissue-based ELISA, as evidenced by these analytic response curves, and really immunohistochemistry and next generation immunohistochemistry rocks. And don't be a stranger. I'm here to interact with you. with
Dr. Rusilla has his hand up.
Yeah.
hand up.
Yeah, just a great presentation, Andrew, and great review of importance of immunohistochemistry. You said very well, immunohistochemistry is a science. You said that several times. And that's why, in 2015, we actually raised the need to have in our lab, as part of our lab team, a PhD trained professional who actually would help with the introduction of new antibodies. This is how Maria Faruqi joined our team, and she brought a tremendous advance, in the quality of the antibodies and the introduction of new antibodies. I have a quick question. With the progress of NGS, a lot of the mutations that are targetable are now studied by NGS, and they're identified by NGS. However, what is the practice in your lab? Because what I prefer to do, at least in cases of lung or thyroid, to give a couple of examples, I still prefer to start with antibodies that I know targets, mutations that are mutually exclusive. And because of that, I don't need, and I save money, by, not having the need of NGS to be done in the second step, and only go to the second step afterwards. So what is the practice in your lab? How do you approach this?
Frequently asked question. In the NGS world, what is the continued relevance of immunohistochemistry? And folks always want to pit me against the molecular pathologist, and the techniques are so complementary. And I'm saying that next generation immunohistochemistry is really surrogate for molecular genetics, and there's no simple explanation to your question, because the answer is analyte specific. And there are and there are applications where immunohistochemistry has, in our practice, replaced NGS or FISH. And the best example of this is one of the next gen IHC markers that I had the privilege of participating in introducing to the pathology community are immunohistochemical stains for synovial sarcoma. So there we have SS18 SSX and SSXC terminus, and the combination of those two antibodies is 100% sensitive and specific for the diagnosis of synovial sarcoma, demonstrated in our initial publication and then in a dozen publications after. And so now H&E diagnosis of synovial sarcoma, application of these two antibodies, and no need for FISH, no need for NGS. Additional advantage of we use gentle decalcification in the bone marrows, compatible with in situ hybridization, chromogenic in situ hybridization, compatible with NGS. But in Surge Path, we've been struggling to flex to an EDTA based gentler decalcification. So we can't do, in a decaled specimen in Surge Path, can't do NGS or FISH. So another potential diagnostic advantage of immunohistochemistry relative to FISH or NGS. BRAF's another example. BRAF immunohistochemistry, we use half a dozen different ways. And in thyroid, if a tumor is BRAF V600E positive, or right now we're working on RAS Q61R in that tissue type with has to be carefully optimized and validated, but with an accurate assay, you can again obviate the need for NGS. We use BRAF immunohistochemistry in colon, specifically in MLH1 PMS2 deficient colon cancers to try to begin sorting out whether they're sporadic MSI high cancers versus Lynch. And the readout, so the pathologist readout, look through the microscope, positive, negative, for BRAF immunohistochemistry in colon is challenging. Much, much, much more challenging than in thyroid or brain or histiocytic neoplasms. And so the readout is almost always positive or negative, but sometimes it's equivocal. And so for that reason, and because we're interested in other molecular genetic information, uh, we do BRAF immunohistochemistry and send for NGS. But even when we send for NGS, we have the BRAF result the same day, and we have the NGS result in 10 days or 2 weeks. So and everybody knows this, a key advantage of immunohistochemistry around, and Emil, you're suggesting for triage purposes or triaging additional testing is you have the result so quickly.
Yep.
Dr. Nielsen, you have any comment?
Of course I do, Liam. You know I do. Andrew, nice to meet you, Andy. So I'm a molecular and translational anatomic pathologist. So Emil's question always perks my ears up. I like what you kind of answered on that question because I would agree that they're complementary. I would warn Emil and trainees and others on the lab on the line that mutual exclusivity is still a statistical tendency. And when it comes to therapeutic planning, I would not use any single analyte of mutational status readout to say, okay, we can give predictive information in terms of therapy planning. But Andrew, and this is my question to you, I think that what you were indicating is very strong, right? Which is IHC and the primacy of it, particularly when we talk about lineage determining factors, transcription factors. I think it is probably still the absolute platinum standard in terms of making a diagnosis, understanding differentiation. I'm wondering if you've done kind of studies of all of the different factors that you have developed and compared that to, carcinoma of unknown primary classifiers, things like that. Because my role, I get people asking me, hey, the IP, the immunohistochemical profile shows this, the gene classifier shows that, and as the molecular pathologist, I always believe the IHC profile, but I'm wondering what your data is on that.
Uh, you may not be surprised because I said that I'm a general surgical pathologist, or I suggested that I was very broad, and I have this academic identity as an immunohistochemistry guru. I alluded to the fact that I'm a neuroendocrine pathologist as well, but I'm the because it's hard. I'm the cup tumor of uncertain lineage pathologist. I'm the person that speaks in that space, and because because our lab is so strong, and because uh, our and so extensive, and because my position is so strong, that we do very little send out of gene expression profiling for carcinoma origin or tumor of uncertain lineage, cancer type ID being one, like sort of first in, primacy in terms of they were sort of first commercial. Uh, I so the clinicians trust me and us more than they trust the gene expression profiling. I lecture on this specific topic, and unfortunately, like locally, I can only speak anecdotally. Uh, they they consider I consider the test I consider the test to be complementary to immunohistochemistry, and I consider it to be hypothesis generating. But I have numerous anecdotal instances, and it's usually the cases that we send out are the cases that our group and I can't solve, and it's very rare. It's like 2 or 3 cases a year, where we're just and I'll actually let me put a pin in that because there's an answer. Uh, and the results that come back are I can't recall a single instance in which I went, oh, that was a diagnosis that I missed. The the diagnoses that come back are often manifestly incompatible with the morphology, with the anatomic presentation, with the molecular genetics if it's gene expression profiling. If we do it, we evaluate it and we evaluate it in the context of the total case. Uh, NG, there have been, and this is continues to be an area of extensive study, and these commercial vendors, it's a great idea, uh, but uh, and they're highly motivated to demonstrate the utility of their tests, but there have been several clinical trials that have been performed using a carcinoma of unknown primary tumor of uncertain lineage classifier to select patients for chemotherapy versus physician choice of cup chemotherapy, and the clinical trials are negative. So so far, these tests have not demonstrated, and it's a high bar, right? Like in the context of a clinical trial, improved patient outcomes. One for the trainees, especially for faculty in the trenches. If I was going to give the cup tumor of uncertain lineage talk, I always say age, gender, anatomic location, and Andy, this is the advantage of, this is why IHC wins. The problem with the gene expression profiling is it divorces it from all the context that gives us our superpower.
For
Uh,
sure.
age,
I'm
gender,
glad we're aligned.
age, gender, anatomic location, and personal history of malignancy. So if a patient has a personal history of malignancy, no matter what the tumor looks like or how it stains, the anchor for your diagnosis, you need a really, really, really incredibly strong evidence of an alternative diagnosis. The anchor of your diagnosis is that it's an unusual morphologic or immunophenotypic presentation of whatever that primary tumor was. And what has become abundantly clear over the last 5 to 10 years, really transformative, was
Great.
the initial description and now demonstration that this is a pan-cancer phenomenon of the frequency, the pervasiveness of SWE/SNF subunit inactivation, pan-carcinoma, I should say. So in this carcinoma of unknown primary tumor of uncertain lineage talk that I would give, I say everything de-differentiates. So if it's screwy, these cases that we would send for cancer type ID, now they're frequently SMARCA4 BRG1 inactivated, deficient, undifferentiated carcinomas or de-differentiated melanomas, but you can have de-differentiated anything.
Yes, it's
Uh,
it's wonderful, wonderful discussions.
Yeah, unfortunately,
I
we
know
have.
people still have questions, but unfortunately, we're running out of time.
Yeah.
Amy is going to send everyone an email link so you can ask Dr. Bellizzi if you have any more questions. Thank you, Andrew. It's for this wonderful, highly informative talk.
Yeah, to
And I'm
Ismail.
sure we're going to continue consult your expertise. Emil?
No, I just want to say to Ishmael and Khalil, I can share his, Andrew's email, and they can ask him directly the questions. Thank you.